Cosgrove, and this is Mission Unstoppable. Coming up,
we're going to open it up and reveal this fossil that hasn't seen the light of day for 68 to 70 million years.
Erica digs deep for this story on excavating dino bones. Then, struggling with dyslexia?
Students who struggle to read might fall behind in school, even in subjects they'd otherwise love.
And your eyes won't believe what these filters can show us.
We found traces of water in the atmosphere of a planet light years away.
Plus, you share a lot of things with your pet. Are diseases one of them?
We don't want to scare people away from interacting with animals. It's actually usually quite safe.
Get ready to meet the scientists, inventors, and heroes who help make our world a better place. The future is here. The mission, unstoppable. Our first scientist literally rips ancient bones out of rocks. If you don't think that's cool, I really don't know how to impress you. Erica has more.
The coolest bones are always and forever dinosaur bones. Look at them. But what does it take to actually pull fossils out of solid rock? To find out, today I'm meeting with Myria Perez, a paleontologist at the Perot Museum in Dallas, Texas.
I am a fossil preparator, which means I get to release dinosaur bones and other fossils from rock.
So you're actually preparing them. You're working on them before they end up in an exhibit.
Yes.
And what are you working on right now?
We got a new jacket from the field of what we think is this dinosaur, Pachyrhinosaurus.
Okay, what do you mean by jacket?
So fossils come from the field to the museum, and we got to make sure that they're supported and that they're safe when they're traveling, because they are very fragile.
Sure.
So a jacket is a plaster and burlap case that surrounds it. You'll be able to cut one open when we go inside. Shall we go to the lab?
I would love to. This is a jacket, and we're gonna open it?
We're gonna open it and reveal this fossil that hasn't seen the light of day for 68 to 70 million years.
To open the jacket, we need special respirators to keep the plaster out of our lungs, eye protection, power tools, and gloves. While Myria does the hard part, I get to help with a very advanced piece of lab equipment, a vacuum, to make sure plaster doesn't get everywhere.
We have opened the jacket. We'll take a photo to make sure we've got everything in context.
By context, she means that paleontologists keep track of exactly where the fossil was embedded in the rock and where it was in relation to other fossils. This can give them valuable clues about the type of animal, its environment, and even how it died.
And then we take small tools and we just brush it off and take away the dirt. You actually start using what's called an air scribe.
The air scribe is a special tool that's kind of like a mini jackhammer. Myria maps out a safe area for me to scribe, so I can't mess up that much.
I feel a little bit nervous knowing that I'm about to actually work on something that contains millions of years of history.
And removing the fossil from this rock could take literally weeks.
It's gotta take a lot of patience to do this.
I only have patience for this, and that's it.
After you've done the exacting work of removing the fossil from the rock, it's time to figure out a dinosaur-sized puzzle.
So let's go take it to our skeleton and figure out where it belongs.
Okay.
So I'm thinking it's probably gonna be from the skull area. So with this piece, I really think it's part of the jaw. It's probably the lower dentary.
Oh, yeah?
And these are the tooth sockets, so where the teeth would be.
You're right.
This is the right dentary.
Right dentary. That means jaw. So we have sort of like a prepared, finished fossil here. You compared it to this skeleton to figure out where it might fit on the Pachyrhinosaurus. What do you then do with that?
So it'll either go on display later, and it'll most definitely go in collections where it'll be researched by scientists all over the world.
Wow. So they'll be using that to sort of figure out more about history.
Yes.
And you're just around all of these all day. Like, this is a room full of history, and you actually work with it.
Whenever you get kind of in a rut, or if you're working on something that's super fragile and you get a little frustrated, I always come out here and just imagine these things living and breathing, and it humbles you.
So next time you see a dinosaur in a museum, remember the skilled scientists like Myria who pulled those secrets from solid rock.
In my dressing room between takes, I like to check in with our experts. So I asked Myria what she's been up to since we last filmed. Check it out.
Hi, Miranda. Since we last talked, I have moved to Washington, D.C., and I am still working with fossils that are millions of years old. I'm also learning how to do something called scientific illustration with a special tool called a camera lucida, which allows me to accurately capture all of the details of specimens such as shark's teeth and even T-Rex teeth. If you've ever looked into a science textbook or even a scientific paper, you might have noticed these illustrations. It's very important these drawings are really accurate because other scientists and researchers can actually use this drawing to study and use in their research. Scientific illustration is the perfect mixture of science and art. That's what I really enjoy. Although my fossils aren't going to run away from me, I do need to get back to the drawing board. Bye!
Dyslexia is hard to spell and even harder to understand. Good thing Dr. Brain's here to make it all make sense.
When we hear the word dyslexia, we often picture typed words and sentences with their letters out of order. But people with dyslexia don't just process written words differently. Their brains have different pathways to process all kinds of information. How is this possible? Well, the answer, of course, is in your brain. Hello, I'm Dr. Brain. Okay, my real name is Crystal Dilworth, but I have a PhD in neuroscience, so it's just easier to call me Dr. Brain. So, I'm Dr. Brain! What is dyslexia? Well, it starts with sight. You thought I'd say your brain, didn't you? Well, I know at least 12 other body parts. Photoreceptor cells in the retina of our eyes receive the images of the text, and this information is transmitted along the optic nerve to the visual cortex. But your brain doesn't really have a set reading section, so the true meaning and sounds of the words it sees still need to be decoded. Our brains didn't evolve to understand written language, because we were too busy with the whole walking upright and controlling our vocal cords thing. Understanding written language requires a variety of different brain regions working together. Basically, one part of your brain looks at some scribbles that looks like C, A, and T. Then a different part of the brain puts them in order and recognizes the word cat. Then a different part knows what that word sounds like in spoken language. It's a real team effort up there. Woo! But dyslexic brains are organized differently. The way dyslexic brains work is optimized for making big picture connections, but at the expense of processing fine details. So while dyslexic brains might struggle with reading or writing, they might be great at spatial reasoning, like 3D art, design, engineering, or even organic chemistry. The abstract and complex concepts at which dyslexic brains excel aren't introduced in school to students until they're older. So in early grades, dyslexics often struggle. So much of our education is based on communication through written symbols, reading, arithmetic, history, science. All of that is given to students in books and worksheets and tablets. So students who struggle to read might fall behind in school, even in subjects they'd otherwise love. But with time and care, many students who struggle with dyslexia as kids can find academic and professional success. And when they do, they'll be in good company with scientists like Albert Einstein, famous writers like Agatha Christie and F. Scott Fitzgerald. You can't outgrow dyslexia, but there are techniques and ways to help people mitigate and adapt to the challenges. Even technology, like text-to-speech and special fonts available on websites, e-readers, or tablets, can be a big help. So if you're someone with dyslexia, know that it isn't a weakness or a mistake. Your neural pathways just may be wired a little differently, like millions of other people. And I'm gonna tell you a secret.
There's no such thing as normal.
Okay, bye!
How does a space telescope take some electromagnetic radiation and turn it into the coolest photo you've ever seen in your life? Let's find out from Astro Athena.
Look at this photo. Pretty incredible, right? This is the Pillars of Creation, part of the Eagle Nebula, where masses of gas and dust gather, condensing together to become new stars. But this stunning vista isn't something you can see with your unaided eye. This is what it looks like with your regular old peepers through a telescope. So how did a camera capture this? I'm Athena, your friendly neighborhood astronomer. And today I'm going to show you how science and technology come together to make incredible images like this possible. While stars send out electromagnetic radiation across the visible light spectrum, they also produce electromagnetic radiation outside of that spectrum. That's invisible light we can't detect because of our normal human eyes. Shorter wavelengths of light beyond our visual capacity are called ultraviolet, or UV rays. The other side of the spectrum, we have longer wavelengths called infrared. So how do we turn UV or infrared images into something we can see and study? There's a lot of science behind this, and one of them involves filters. Right now, this flashlight is white, which is a combination of all the wavelengths on the visual light spectrum. But this filter only lets wavelengths of orange and red through. Scientists use a similar technique on their telescopes. For example, the James Webb Space Telescope has more than 24 filters, and it can look at the same area over and over, each time using different filters to capture light for a very specific wavelength. NASA image experts and scientists assign colors on the visible light spectrum to these images of UV or infrared waves. For example, if they capture light that is between 5.6 to 11.3 microns, they can assign that as the visible color blue, or assign light waves between 18 to 25.5 microns to red. These individual photos are then composited and woven together into a final stunning image that scientists can process with their normal human eyes. This can be adjusted to help the cameras look past dust and clouds to find exactly what they want to study. They can even help the telescope study distant exoplanets as the planet transits in front of other stars. Using filters to look for H2O specifically, we've found traces of water in the atmosphere of a planet light years away. This exoplanet is known as WASP 18b. That's incredible, and definitely not a planet full of wasps. So many groundbreaking technological advances have to work together to let us explore space and time. But no matter how advanced the technology gets, it's all based on simple principles of physics. And they make it look so cool. Just look at it. I'm Athena, and always remember, per aspera ad astra. Bye.
We can get a lot of things from animals. Hugs, licks, eggs sometimes. But we can get some bad things as well. We sent Nabil to learn more.
Humans and animals have been living side by side since the dawn of creation. And over time, we've learned to adapt and even enjoy each other's company. But sometimes these furry little creatures can get us sick through no fault of their own. Don't worry, buddy. I know you'd never do it on purpose.
Today, I'm in Palo Alto, California, to meet with microbiologist Dorothy Tovar, who is studying why some diseases make humans sick, but not animals.
From the foods that we eat, like cheese and kombucha, to infectious diseases, there are tons of ways that we interact with microbes that are really important for our lives. And what I am really excited about is how we can use this knowledge to improve our daily lives.
Dorothy studies diseases that transfer from animals to people, called zoonoses, or zoonotic diseases. Zoonoses are caused by bacteria, viruses, parasites, or fungi, and include dangerous diseases like the bubonic plague or coronaviruses. Dorothy's specialty is bats, but all kinds of animals can share diseases with us. That's why today, Dorothy is challenging me to find different ways that humans and animals interact around this farm. And it starts with an easy one: touch.
Human-animal interactions. Okay, what about horseshoes? Since you put the horseshoe directly on the horse, direct contact?
Exactly.
I'm assuming there at one point was an animal here. Even though it's not here now, I could still potentially get a disease from, interacting with it.
Yeah, exactly. And what you're describing there is called indirect contact. If this water is coming from a local stream or river and is not at all cleaned in some way before we drink it, that is an example of waterborne transmission.
Chickens lay eggs, so if we eat the egg and we get sick, maybe that's a form of transmission?
Exactly, you're touching on foodborne transmission. It's possible to get a bacteria called salmonella from uncooked eggs. There's one more, which is vector-borne transmission, when you come into contact with usually insects like fleas or mosquitoes that can transmit different germs that way.
With all these ways for humans and animals to share diseases, does this mean we shouldn't be hanging out? I mean, me and the donkeys were just starting to vibe.
We don't want all of this to scare people away from interacting with animals. It's actually usually quite safe. So some of the things we can do include washing our hands after petting animals, making sure that we don't drink water that comes straight from streams or rivers, also wearing bug spray when you go outside, not eating any uncooked meat products or things with eggs. So no more raw cookie dough.
Oh, you're kidding.
I know, it's a bummer.
To see if a human or animal has been exposed to a certain disease, scientists run an enzyme-linked immunosorbent assay, or ELISA test for short. This test checks to see if there are bits of specific bacteria, virus, or fungi circulating in the blood. These small bits of a disease are called antigens, and we can detect these antigens using antibodies.
An antibody is a specific protein that immune systems create that target the antigen. And then when it does recognize it, it binds really tightly. And you can think of them as kind of like reporters. They like flag down the disease and then bring other immune cells to come kill the disease wherever it is in your body.
Sounds really cool when you put it like that.
Yeah, our immune system does not play.
Today, we're going to be testing for salmonella using what's called a sandwich ELISA, which gets its name because the antigen is sandwiched between two antibodies. The wells in this tray get filled with different samples. First, some are filled with a sample that we know has salmonella. Others are filled with sterile samples. And finally, some are filled with the substance that we are testing for salmonella. Then, using a color-changing substrate, any well containing the antibody turns blue. So not only does our contaminated control group turn, but any other well that has the zoonotic disease. That's right, I nailed my first ELISA test.
Dorothy, thank you so much for an amazing day. I mean, I learned so much. And you taught me how to prevent zoonotic diseases by washing my hands, which, by the way, I'm gonna go do right now.
Yeah, do that.
Off to wash my hands, goodbye!
Welcome back. Before we go, we have one last thing.
Get comfortable with carving your own path. It's totally okay if you're the only person who looks the way you do or is interested in the things that you're interested in.
Mentors will do you so much good, and it is an amazing relationship that you can have with somebody for a lifetime.
That's it for this episode of Mission Unstoppable. Next week, we're putting together a story on intelligent blackbirds, and the critics are just raving. See ya! If you're watching this, you must have really liked the video. Make sure you follow and subscribe, and check out these other videos that are even better. No, really. I've seen this one over 100 times.